Thanks everybody for the opportunity to present here today. I think I'm what's keeping you guys from the grand opening in the exhibit hall, so I'll try to keep it pretty simple. So a little bit about Demeetra. We're actually located in Lexington, Kentucky, not Massachusetts, but we're known for horse racing and of course bourbon. That might have drove all the hands-on research we do. Maybe not, though. But most of Demeetra is hands-on research. So a lot of us are PhD scientists, and we're continuously working on all of these technologies to optimize them and to transfer these technologies to our clients so they can easily use these and be successful as they go through their evaluations of the technology and become quicker to success. We also offer innovative commercial licenses that don't have that complexity and burdens with royalties and milestones. So a little bit about Demeetra's core technologies. Our first one is Harbor N transposase. So this is based off of the piggyback transposase. And we all know that transposases, like piggyback, can integrate cargo at highly expressed sites, like open chromatin and non-coding regions. And they're also notorious for the large cargo capacities, so up to 200 kb. In-house, we've done 183 kb, and that's a bacterial artificial chromosome, although I wouldn't recommend it because it's very difficult. So our second technology is our Cas-CLOVER nuclease. This is a highly efficient, targeted dimeric nuclease. So it differs from the monomeric CRISPR-Cas9 that is typically out there, and you still have that ease of use. So you can use the guide RNA plug-and-play infrastructure, same types of orientations. And the Cas-CLOVER utilizes an inactivated Cas9 molecule, and it's fused to our proprietary CLO51 endonuclease domain. And this is this creates a high fidelity system which lacks off targets and translocations. And CLO51 is a type 2 restriction endonuclease, so it actually cuts like one too. So it's an obligate homodimer, and it needs that dimerization to actually take place for the cutting to actually happen. So it creates these large deletions with these overhanging staggered cuts, and this is leading to higher knock-in efficiencies. So we produce recombinant protein in-house, and we can test the cutting mechanisms of Cas-CLOVER, and we can compare it to Cas9. So this is if I can get the pointer to work. Okay. So we typically call this the left side and the right side for Cas-CLOVER. This is what the left guide binds and the right guide binds, and it brings this complex in together, so then CLO51 can actually cut. So we would take genomic DNA, you could do PCRs from CHO cells, mammalian cells, and then mix that with synthetic guides with the protein. And then you can actually look at the end products. And so here with Cas-CLOVER, it's cutting in between the left guide and the right guide, and you see this nice 4-base overhang. And with Cas9, this is very typical of Cas9. It's 3 base pairs from the cut site, and it's a blunt cut mechanism. We're seeing improved efficiencies with Cas-CLOVER compared to Cas9 in cell line development, cells like CHO cells. So we're targeting the famous glutamine synthetase. With Cas-CLOVER, we can see around 75% cutting efficiency using a variety of different guide RNA scaffolds. So you can do the 2-part guide RNA, where that's a complex or hybridizing the tracer RNA with the crimp RNA and then transfecting, or you can use the single guide RNAs from IDT and Synthago. So very, very consistent 75%, and then Cas9 showing around 50–55%. We're also seeing improved knock-in efficiencies, and one hypothesis is that staggered overhang cut mechanism that Cas-CLOVER is making is improving and bringing more of that homologous recombination machinery to that target site to actually increase the knock-in efficiencies. So with Cas-CLOVER, this is a 7 kb knock-in. This is in HEC293 cells. and this is with a GFP plus puromycin. So it's around 7 kb in size. Post-transfection, without selection, we're seeing around 10% with Cas-CLOVER. With Cas9, it's around 3%. After a little bit mild selection with the puromycin, we're able to increase that gene-edited pool to around 70%, while as Cas9, that pool is only reaching around 30%. So we're utilizing Cas-CLOVER to build commercial cell line platforms. One system that we're using as the parental is a CHO K1. We're adapting it to suspension. And I'll have to give a shout out to Valeria Steffi. She's our Senior Director of Cell Line Development. So her group has done a lot of this next stuff that we show. So the adaptation took place, it's in suspension, and now we're creating a GS knockout. So recently, we all know there's a glutamine synthetase gene in CHO cells. It's very famous, chosen, and things like that. But there's also a pseudo gene too. So we wanted to knock out both of these biallelically. We were utilizing Cas-CLOVER because we can actually target these at the same time or sequentially. Sorry about that. We can do this sequentially as well. So the blue guides right here will target only the pseudo gene, and it won't cut chromosome 5, the main gene, glutamine synthase gene, because there's an intron in between. So the space is too far away for Cas-CLOVER to actually cut. The red pair will work on chromosome 5, but it won't work on the homology on the pseudo gene because there's a lot of mismatches in one of those guides. So when we do a T7 assay, we're actually taking the blue guides on chromosome 1. You can see nice cutting products from that guide, but no cutting products from chromosome 5. And this is the same case if you took the red guides and used them for the other targets. So with Cas-CLOVER, we're seeing indels that are very large. So typically, CRISPR-Cas9 will give you 1 to 6 base pair deletions in general. But for Cas-CLOVER, we're seeing bigger deletions, more noticeable deletions, easy to characterize deletions. So these can be 16 base pairs for one clone, another clone had 26 base pairs, and another clone had 28 base pair deletions. And this is really giving us a really nice glutamine sensitivity phenotype. So with glutamine, these cells are behaving really nicely. Now when you remove glutamine, these cells are dying within 9 days. So, and that is a benefit of having both the main glutamine synthetase gene knocked out as well as that pseudo gene. So utilizing the CleanCut GS CHO cell line as our parental line, now we want to make a suite of different lines. So to expand on this CleanCut GS CHO cell line, now we want to make a Foot8 knockout. So we want to produce afucosylated antibodies so they have enhanced ADCC. Our cutting efficiency with Cas-CLOVER is very high for Foot8. We actually found really good guide pairs that target exon 9, and we're seeing around 80% cutting efficiencies. So a large portion of our pool is already edited. Whereas Cas9, it's around 55% again. We can do LCA selection, so that's lens culinaris. That's going to bind to those fucosylated glycans on the surface, and then the cell dies. So after selection, we have a pool that already is 98.5% Foot8 negative. And then we can sort them from staining with LCA and get 100% pool, and then from there we can single cell clone. Here's our frequency of distribution of indels. I mentioned this previously, showing that the indels for CRISPR-Cas9 is largely going to be in this region, whereas Cas-CLOVER is really spread. So we have clones that have full exon deletions. We have multiple clones that have half exons deleted, and this really makes a disruptive knockout. A really good thing is now we want to test, since these are in the parental glutamine synthetase knockout, we want to check glutamine synthetase, or glutamine sensitivity. So a lot of these clones, again, with glutamine, they're behaving just as normal, and then they still have that glutamine sensitivity. So this is really nice to see because they're not adapting to growing without glutamine, and they still have that functional glutamine knockout, and this means that they're metabolically clean and they're well controlled. So in addition to the foot 8 cell line from the parental CleanCut GS cell line, we're also looking at host cell proteins. So host cell proteins often affect a lot of downstream applications as well as upstream. So they affect the purity of your protein, they can degrade your protein in the supernatant, and they can affect the downstream purification processes. So here we targeted 3 different host cell proteins. We utilize a couple different pairs targeting these just as an initial test, and we're able to identify the best pair for each host cell protein. So now we can take each of those pairs and perform a multiplex. And we utilize the Lonza nuclear effector, but we also use the neon electroporator as well, but most of our stuff revolves around the nuclear effector. Once we do the transfection, now you have a pool of edited cells. These could have a variety of different combinations of those host cell proteins knocked out. There's no selection, so we utilize a single cell cloner. This is the VIPS. So now you have single cell clones in a plate, and we can utilize a multiplex PCR and a variety of different methods to come up and identify which of these single cell clones have a single knockout, a double knockout, different variations of a double knockout, as well as potentials for a triple knockout. And some of these we're seeing 6 to 7 alleles edited, which is kind of bodes to, you would think, 6 alleles for 3 genes. CHO cells are alloploidy, so you do see some variety there. So we are seeing 6 to 7 alleles knocked out at once. And then we can look at the protein expression. So how are these gene edits correlating to the actual protein production of these host cell proteins? So this is our regular control. This is our CleanCut GS cells showing those host cell proteins, and then how all of our clones are expressing that protein. So a lot of them are knocked out, a lot of them show a little decrease, maybe by half. So we're picking the ones that have full protein abolishment. So now to proof of concept for these host cell proteins, we use the Harbor-N transposase to stably integrate these into this cell line. We have a variety of different clones that we're doing this with, so it gets complicated. But we're producing just a regular IgG as well as a masked IgG. And these have cleavable linkers for a nanobody that these host cell proteins should not be able to work on. So after a 24-well, deep well fed batch, you're able to clean these up with protein A and then look at what type of product that you're getting. And this is very early stages, but our initial test for one of the clones is that it's able to produce that IgG perfectly well, and it's also able to produce that masked IgG, looking at heavy chain, the masked light chain, a little bit of cleavage from that light chain, and actually there's 2 distinct bands here, and that's because there's 2 sites for cleavage on that light chain. So you can actually see those species in this gel, and you can also see the mask as well. So majority of this is a fully functional antibody, but there is still some cleavage because this mutant in particular is not fully knocked out. But it's nice to see that we can actually analyze these. So we're also using Harbor-N transposase to make stable trastuzumab expressing cell lines. And the Harbor-N transposase is license free. So there's no royalties, there's no license to pay. We just sell it as a reagent. It's an optimized mRNA. We also provide a bunch of transposons that you can put your gene of interest in, or we have a transposon that actually is what made these trastuzumab expressing cells. So really nice kind of plug and play to maybe your cell line of interest, or you can buy our commercial CleanCut GS cell line and kind of check this out for yourself. But for an unoptimized 24 deep well fed batch, our viable cell densities are only at around 10 million cells per mil. But as you can see, our top clones are reaching over 5 grams per liter. The most interesting thing, in my opinion, is our cell-specific productivity. Most of our clones are 60 and above for cell-specific productivity, and this one's around 110. So this is a really high specific productivity, and I think this bodes pretty well for a transposase type of system. So to go further into our transposase system, we are using an integration-only expression cassette to really get an idea of the activity of our harborin transposase, and we're comparing it to one of the leading transposases, super piggyback, in cell line development. So the standard transposon has these anti-parallel ITRs, you have core insulators, and then your promoter driving EGFP. So then once super piggyback comes in, it'll actually grab these ITRs and then put that into the genome. But you can get transient expression. And transient expression really kind of inhibits real-time quantification of how active your transposase is. So now we're utilizing an integration-only transposon cassette. So this is actually switching the ITRs in a parallel formation. And what happens is your EGFP is in a different orientation than your promoter. So you don't get any of that background unless it's repaired correctly via piggyback and the repair mechanism, and it actually turns on EGFP if it's incorporated into the genome. And this was published in 2020 in a Neuron Cell report. So we're comparing this to super piggyback and CHO-K1 suspension cells. You can see the mock has no GFP expression. The plasmid only has no GFP expression, as it should. And then we're using super piggyback with a variety of different amounts of that integration-only cassette. And you can see a nice stepwise amount of GFP expressing in these CHO-K1 suspension cells. And there's not much difference with harborin either. Even though super piggyback is a form of a hypervariant, harborin is performing very well in cell line development types of cells. The real difference is in T cells, harder to transfect cells like iPSCs, HSCs. Super piggyback definitely exceeds harborin in that context. But for cell line development, harborin is a very useful transposase. So altogether, Demeetra has a very comprehensive freedom to operate cell line package. We have, this is busy, but it's simplified. We have a license with the CVC that covers the guide RNAs, as well as the usage of those with an inactivated Cas9 molecule. Poseida is our sister company, and they can use Cas-CLOVER for cell and gene therapy, and Demeetra has that exclusive license for cell line development, bioprocessing, as well as agriculture. CLO51 is owned by Helmholtz Munich, and Demeetra being an R&D and gene editing company, we're also continuously making improvements to this entire system, and we also have patents that has been out for a year or two on some new variants. But all of this is bundled into one simple package that you won't have to deal with any outside licensing structures, no royalties. So it's very nice and something that you can't get going to the CVC alone or going to the Broad Institute alone. So now that covers Cas-CLOVER, Since the CleanCut GS CHO cell line was made with Cas-CLOVER, this instantly gives you access to this cell line without any other hassles. And then Harbor In, it's license-free, it's just a reagent. So it's a really nice cell line development package so you guys can make your antibodies of choice and be successful. With that, it's exhibit hall time. I will take questions for sure, but come visit us at booth 216. And then Christian Cummings, one of our business development managers, is presenting a poster number 25. And with that, I will take questions.
Thanks so much for the talk. It's really cool. I'm curious about the overall efficiency of your triple knockouts. Kind of what fraction of your clones had all 3 of those deleted at both alleles? And then did you compare that efficiency against a normal Cas9 system? And is it different for your Cas-CLOVER?
We didn't compare it to the CRISPR-Cas9 system just because most of our work, we're kind of figuring out that this is one of the host cell proteins is a concept that we're working with right now. We're still interested in that. But with the host cell protein work that we're doing, I think the percentages of the clones that we looked at, it was above 50 to 100 clones. And I think about 20% were all triple knockout. You're right here. I can hear you.
Okay. So when you were doing the host cell protein knockout, it looked like you had a couple that actually increased, or is that some sort of marker? The couple over on the right, and if it was that, any idea why?
Yeah, I'm not sure about the potential increase. It was too close to tell. I didn't put the normalization on there, but when it was normalized, it wasn't. It was 1.1. 1.2 normalized, so not too much of an increase.
Any other questions? Okay.
If it were increased, it could be a compensatory mechanism as well. A lot of these host cell proteins are promiscuous, so.